The final laboratory skill we will discuss is the preparation and evaluation of the stool or feces. This is done to identify parasites that might be within the dog or cat's body. Because most parasites or worms attach to the intestines, the adult worms remain in the intestinal tract, shedding only eggs in the feces. For this reason, we must look for the eggs rather than the adult worms. Unless the owner brings the adult worm their pet past, you must look for the eggs. Such eggs, though not shed all the time, are shed a great deal of the time. The technique most often used to get a concentrated sample of these eggs is called a fecal flotation test. The first step involves obtaining a workable stool sample. If the pet owner did not bring a stool sample with their pet, you can collect one from the pet's cage or directly from the pet with the aid of a fecal loop. This loop should be lubricated and passed 1 to 4 inches into the anus, being careful not to be too forceful or cause the animal any pain. This procedure is often uncomfortable and will require some restraining assistance from another hospital staff member. When passing the loop into the large colon, you will likely feel the slight resistance of the stool. When the tip of the loop is likely embedded into feces, give the loop several twists and remove. If the stool is not present, you may attempt this procedure again slightly deeper if the pet was not uncomfortable. A smaller fecal loop is available for obtaining samples from cats and small dogs. We are now ready to proceed to the next step. Mix a small quantity of feces with dense flotation solution that contains the salt sodium nitrate. This concentrated salt solution is used to top off the sample once it has been well mixed. The final solution is covered with a cover slip once it has been filled to the very top. It is at this time that the eggs will rise to the top of the solution and will come to rest in a position against the cover slip. It is recommended that 5 to 10 minutes elapse to give adequate time for the eggs to float up. Now carefully lift the cover slip and set it on a microscope slide, being careful to transfer some of the contents of the fecal solution adherent to the cover slip. If a microscope slide is used to cover the fecal solution, you will need to quickly flip it over before placing a clean cover slip on top of it. If excess solution is on the underside of the slide, it should be wiped dry so as to not get this salty solution on the microscope. The other technique used for detecting intestinal parasites is a direct smear. With this technique, a small amount of feces is spread onto a glass slide. A drop or two of saline is added and stirred lightly. Place a cover slip over this mixture and examine with the microscope as before. This technique is especially helpful in diagnosing certain protozoan like Giardia. Always follow good hygienic practices and thoroughly wash your hands after touching containers with feces in them. You are now ready to examine this slide under a microscope. Before discussing what to look for in identifying parasites, it may be helpful to briefly overview the components of a microscope and how to operate this precision instrument. The light source is turned on and adjusted to whatever brightness level is indicated when looking through the microscope. The stage is the platform on which the microscope slide is secured. The slide can be moved about by adjusting 2 knobs below the stage. One moves the slide horizontally, the other vertically. Most microscopes are equipped with 3 or 4 objectives, or lens. These lenses determine the magnification. The objectives range from 4x to 100x magnification. When looking for parasite eggs, you will generally start out scanning the slide with a 4 to 10x objective in place. When working with a microscope, be careful not to touch the lenses and only clean them with special lens paper, not ordinary tissue products, as these may scratch. You will probably need to first adjust the eyepieces on a binocular microscope to a distance that allows you to comfortably look with both eyes. When peering through the microscope, it is helpful to relax your eyes and focus them as if you were looking at something on the counter below. Once the slide and 10 times objective are in place, you are now ready to find the correct focal distance. Once the slide and 10x objective are in place, you are now ready to find the correct focal distance. There are 2 focus knobs on a microscope, a major focus and a minor focus. Start with the objective close to the slide and slowly increase the focal distance using the major focus knob. When any of the contents being examined become visible, you can switch to adjusting the minor focus knob until the image is clearly in focus. As the slide is repositioned on the stage, you will most likely need to make adjustments to this minor focus in order to keep the image crisp. With one hand, you will likely need to readjust the focus, while with the other hand, you will need to control and move the slide. By starting in one corner and working a pattern like that demonstrated here, it is possible to not miss eggs. At each location on the slide, you will need to stop and quickly scan for the presence of parasite eggs. If nothing is found, advance to the next field until the whole area is examined completely. Identifying parasite eggs will require time and experience. We will introduce you to the most commonly seen parasite eggs in this program, but these are not by any means the only ones potentially present in animal feces. Air bubbles, pollen, and plant material all may resemble parasite eggs at times, so if you are ever in doubt as to what you are looking at, consult your veterinarian or trained technician for their assistance. The roundworm egg is the largest of the common parasite eggs and possesses a thick, dark wall. Roundworm eggs are generally easy to identify because they are large and rounder than most worm eggs you'll see. These eggs are commonly found in young puppies and kittens because these worms are passed to the newborns through the placenta before birth or in the milk after birth. The hookworm egg is ovoid in shape and contains small round cells which resemble a cluster of grapes. Hookworms are a common parasite of young kittens and puppies. The whipworm eggs are quite unusual because of their almost barrel-like appearance. You might remember this unique egg shape by imagining a barrel filled with whips. Tapeworm eggs are contained in the segments of the tapeworms that are passed, so we seldom ever see the actual eggs of the tapeworm with a microscope. This is important because many dog and cat owners will see these worms being passed and bring their pets in to be treated. Upon examining a stool sample, tapeworm eggs are not likely to be seen. Another type of internal parasite, called coccidia, is not a worm but rather a protozoan. These organisms, when noted on a stool exam, will be quite small and ovoid in shape. They may resemble a roundworm egg but are quite a bit smaller. Our discussion in this program will be limited to identifying these most common parasites. Diagrams of these common parasite ova can also be found in your workbook. As mentioned at the beginning of this program, there are many other laboratory skills you will inevitably be taught. Your introduction to working with blood, urine, and fecal samples should, however, provide you with a necessary foundation to help you be a valuable asset in uncovering the mysterious diseases and problems many of the pets may present